what we can see is this. Let's, once you complete that, what we see is that there's going to be sp2 sigma bond that's going to generate electron density here, right? And then we have a lone pair that generates electron density here, another lone pair that generates electron density here, here. And these are all paired, right? And so we got a pair here. Pair here in the bond of lone pair, lone pair, lone pair. All the electrons are paired up. And then we have a pi bond. The pi bond is going to have electron density up here. And this is side view. And electron density down here. And the pi bond here is, again, it's only going to have 2 electrons. Some people have 4 electrons in the pi bond. They aren't. There's 2 electrons. Those 2 electrons can be, 1 electron can be in this lobe, 1 electron in this lobe, or 2 electrons in this, none in this, or none in this and 2 in here. It doesn't matter where I put the electron. So on. And so this is the electron density map of O2 using valence bond theory. And in other words, we have the pp pi bond, we have the sp2 sigma, we got the sp2 lone pair, sp2 lone pair, sp2 lone pair, sp2 lone pair, and this is what we have, right? Now, how does that compare to the MO? Well, the MO is, we need to get the, right, so do we use the abnormal or the normal? Normal. We're going to use the normal because the Z is bigger. And so it's going to be sigma 2s, sigma star 2s. And we fill the orbitals just like we would AOs. Sigma 2p, pi 2p. One at a time. The pi's get filled one at a time. Pi star 2p and then sigma star. Sigma star 2p is going to be empty. The pi star is going to have 2 electrons because each oxygen has 6 valence electrons. And so O2 is going to have 12 valence electrons, not 16. O2 has a total of 16 electrons. But remember, I'm ignoring the valence. I'm not ignoring the valence. I'm ignoring the core. So we only do the valence, which is 12 total. This is going to give me a bond order of 2. Now, the way we describe a bond in MO theory is different than the way we describe it. But essentially, it's kind of the same thing. And so here, now this is going to get a little bit messy because there's a lot of orbitals. The first orbital is the sigma 2s. So we put two electrons in the sigma 2s. That's going to give us a bond order of 1. It's like a single bond. So, sigma 2s. After the sigma 2s, what orbital comes next? Sigma star 2s. Now, sigma star 2s is going to have a node. I'm going to draw it differently so we don't get them mixed up. So the sigma star 2s, let's draw with the dash line here. Maybe I drew this too big. This is a sigma star 2s. And this has antibonding electron density. So 2 electrons worth here. Antibonding electron density is going to cancel out the bonding. In other words, if I stop now, do I have any bond here? No, I'd have no bond. Because I have 2 electrons in the bonding, 2 electrons in the antibonding, it's canceled out. No bonding. I need more electron density in the bonding than in the antibonding. All right, now what comes next? Sigma 2p. Now Z is big, so the sigma 2s gets pulled in more than the sigma 2p. And so sigma 2p, I'm going to try a different color here. The sigma 2p gets pulled in less, it's less penetrating, and so there's less repulsion. And so this will be the sigma 2p here. The sigma 2p is bonding electron density. And so if I add in the electron cloud for the sigma 2p, it's going to help, because now I have more electron density. in the bonding region. And so now the bond orders pop back up to 1, excuse me, bond order 1. 2 electrons worth of bonding density here. Okay, after the sigma 2p comes what?
Pi 2p.
The pi 2p's. Now, for the pi 2p's, I have 2 of them, and they're just perpendicular to one another. And so one of the pi 2p's is going to be the side, kind of the side view here. There's one lobe above the nuclei, one lobe below the nuclei, and this is going to be in the plane. And so these are the 2 lobes of the pi 2p. The pi 2p here, it's in the plane, has 2 electrons in that. So there's bonding density here. So that's going to help the bond. And then I have another pi 2p. I have the pi 2p that sticks out, so I'm going to rotate this 90 degrees, so it'd be like a top view rather than a side view here. And for the top view, I'll just use a different line style like this. This zigzag line here. And so this will be the lobe coming out. So this is the pi 2p perpendicular, and then there's going to be a lobe sticking in, which will be dotted here. Lobe sticking out, lobe sticking in. Actually, I could just show you on here. This looks like.
Professor, do you want us to do the little electron density parts of the drawing?
Well, just to get an idea of where the electron cloud is, the overall electron density. All right, so here's the side view, right? Here's the top view. In other words, we're going to have 2 of these. This is perpendicular to one another. And this is bonding density, too. Bonding density. Do you see already it looks different? It looks different because when I look at the valence bond, I only have this, side view here. There's no perpendicular. Do you see? There's no pi electron density perpendicular. Can you see that? Here. And so it's already starting to look different, the electron density map. Because in this case, I got pi in the plane and I got pi perpendicular to the plane, bonding electron density. But what's going to cancel out a little bit of this pi bonding density is what?
Pi star.
Yeah, the pi star, but there's only 1 electron's worth. And they got this mixed up. This is the pi star for this one. This is in the plane. And so the pi star, let's see what color do pi star. Do pi star in, we'll do it in purple? No, can't do it in purple. Black? I'll do it blue. I'll do blue. Okay, so the pi star in the plane, we have 1 lobe coming out like this. Okay, another lobe coming out like this. This is in the plane. The lobe coming out like this. And the lobe coming out like this. Now, we only have 1 electron in that pi star here. And so that's going to cancel out some of the bonding of this pi. And then perpendicular to this one is another one. So this would be kind of the side view. And then if we rotate this one, we get like a top view like one over there. But it's a separate orbital. So how should I draw this one? Okay, the other one, I'll just use blue again. But we'll just make it very thick. And so these are the 2 lobes that are sticking out here, and then there's going to be 2 lobes sticking in. And this is going to have antibonding. density here. Some anti-bonding. So that's going to cancel out some of the bonding. But do you see it's kind of very symmetric? And so it turns out that O2 here, when we look at this, all the electrons are paired here. When we look at this, the electrons aren't all paired. This is going to be unpaired in this, and then we're going to have unpaired in this. And so we have unpaired electrons in here. All the other MOs are paired electrons. And so it turns out O2 is a etc. Anyway, it turns out to be a mess, but if you draw it yourself, maybe it makes more sense. Right? Because you can draw one orbital at a time. But the electron density maps look different, don't they? Let me tell you, for nitrogen, they look the same. You know, for nitrogen, if you do the valence bond analysis and you do the MO, it comes out pretty much the same. It doesn't look all that different. O2 it does look quite a bit different. And so anyway, that's the analysis for this. And so, like I said, MO, you know, MO is going to give us the hybrid at the end. You know, we don't have to figure out what the hybrid is. MO is going to give us a more accurate picture. And so what we're going to do is we're going to get our little pre-introduction to MO. And so we're going to go to the computer lab. But before we go to the computer lab, you know, why is structure so important? So I want to show this old video. Even though it's an old video, it's still pretty relevant.
Systems is remarkable. More than 1 billion species currently exist. We now know that a principal factor in this large number is the billions of ways that 4 rather simple chemicals can vary their sequence in forming DNA genes. We are also discovering that many illnesses can be identified at the molecular level. This provides information for the design and synthesis of molecules, which can be used to treat the disease. If we examine living cells under the microscope, like these plant cells, or these bacteria, or this animal tissue, we can see the movement, growth, and cell reproduction, which are the result of successions of chemical reactions. In attempting to control these reactions, physicians have used various substances, some from nature, and some synthesized in the laboratory. Many of the compounds found to be useful were discovered by trial and error. One such compound was sulfanilamide. Let's demonstrate the effect of sulfanilamide on bacterial growth. A nutrient medium in a petri dish is used to sustain growth of bacterial organisms. These bacteria, harmful to humans, are carefully transferred from a stock preparation. They are gathered in a wire loop and streaked onto the nutrient medium in both compartments of this dish. A small piece of sterilized filter paper is wet with a sulfanilamide solution and placed on the bacteria in one compartment of the Petri dish. The dish is placed in an incubator for 24 hours. During that period, seen here in time-lapse photography, the bacteria multiply and become visible as large colonies everywhere except in the area around the paper disc. The clear zone shows that bacterial growth has been stopped. This inhibition is due to the sulfanilamide from the disc. How does sulfanilamide stop the growth of these harmful bacteria? First, let's examine the function of one of the key molecules that exists in the bacterial cells. It is called para-aminobenzoic acid. Using a geometric symbol retaining the general contours of the molecule, we'll follow its action within the cell. In the cell with the acid is an essential bacterial enzyme. In order for the enzyme to perform its active role, it must first combine with the para-aminobenzoic acid. Only then can the enzyme catalyze the further reactions necessary to cell life. When we examine the structure of the acid molecule and compare it to a molecule of the drug sulfanilamide, we find that they are much alike. This large portion of each is identical. The electron charge distribution in each molecule is also similar, and their general configurations are the same. Using geometrical representations again, let's include sulfanilamide in the bacterial cell along with the normally present acid molecule. The sulfanilamide too can react with the enzyme. But the sulfanilamide not only fails to activate the enzyme as a catalyst, it also prevents the acid from doing so. Thus, metabolism stops, and the bacterial cell dies. The effect of sulfanilamide on cell growth was found by trial and error. But the discovery that sulfanilamide is structurally similar to an essential molecule accounted for the action of the drug. This principle of structural similarity helps us to predict which drugs might be effective against specific diseases. Consider, for example, the use of drugs in the treatment of cancer. By means of tissue culture technique, human cells are kept alive for microscopic observation. Here are normal human skin cells under the microscope. In time-lapse photography, cellular activity appears accelerated. We know of very few differences between the chemistry of normal cells and these cancer cells. One remarkable difference, however, is that the cancer cells grow faster. And another difference is that during the faster growth, the cancer cells use abnormally large amounts of a substance known as uracil. Can a structural modification of the uracil molecule be used to disrupt the fast growth of the cancer tissue? Let's follow some of the steps a biochemist takes in answering such a question. He is familiar with the visible parts of a single cell, like this one undergoing division. We see a set of dark ribbons in the middle of the cell. These dark objects are chromosomes, and they are intimately connected with the process of cell growth. The biochemist has learned to collect chromosomal material from cells and has found that it is composed of deoxyribonucleic acid, or DNA. Because DNA is a large, complex molecule, it is helpful to break it into its component parts and identify them. These component parts can be separated from each other by the technique of paper chromatography. After treatment with an acid, a small drop of the chromosomal material is placed on this large piece of filter paper. The paper is rolled into a convenient shape for the next procedure. We have selected a solution in which the chromosomal material is readily soluble. This is placed in the bottom of a glass cylinder. The solvent is drawn up the filter paper by capillary action. After 12 hours, the solvent front has risen this far. The filter paper is removed and allowed to dry. It is then rolled in the opposite direction and put back in a different solvent. This will ensure a further and better separation of the molecular components. These components are not visible under ordinary light, but if we place an ultraviolet light under the dried paper, we can see that the solvent has distributed the components of the chromosomal material. This distribution has occurred because the solvent front competes with adhesion to the paper for the chromosomal materials, so the components move according to their molecular size and solubility. Thus, different compounds derived from the chromosomes are separated. We'll cut out one of these marked areas and dissolve the material from the paper with another solvent. Now we can analyze the dissolved material with an infrared spectrometer. A characteristic spectrum is produced by each component separated in the chromatography procedure. The absorption spectrum produced by one of these extracts is compared with the known absorption spectrum of the chemical thymine. Apparently, chromosomes contain thymine. Similar analysis of the other components shows that each has its own characteristic spectrum. Identification shows that besides thymine, chromosomes contain adenine, guanine, and cytosine. How do our uracil molecules, which are present in such large amounts in cancer cells, relate to the constituents we have extracted from the chromosomes? We know that these constituents, along with other building blocks, are necessary to make DNA molecules, which in turn have a part in forming the chromosomes. Biochemists have shown that uracil is the substance from which thymine is formed. Without uracil, thymine will not form, preventing the synthesis of DNA, which in turn prevents the formation of the chromosome. When we examine the molecular structures of uracil and thymine, we find that there is but one position in which these structures differ. To convert uracil into thymine, a hydrogen atom here must be replaced by a methyl group such as this. Let's symbolize uracil with its hydrogen atom like this to follow its action within a cell. First, a chemical group called a sugar phosphate residue becomes attached to the uracil molecule. The modified uracil compound then forms a complex with an enzyme. Somehow the enzyme converts the hydrogen into a methyl group to form the thymine compound. This molecule is then freed for eventual synthesis into the DNA chain. How can we disrupt this mechanism and stop cell growth? Clearly, one point of attack is the original uracil molecule. This molecule can be altered in many ways. One of the most promising has been to replace this hydrogen with a fluorine to give 5-fluorouracil. 5-fluorouracil has a number of advantages. First, the hydrogen and fluorine are almost the same size, so that both molecules should fit into the enzyme. Second, the chemistry of a carbon-fluorine bond differs from that of a carbon-hydrogen bond, so that the enzyme might find it difficult to replace the fluorine with a methyl group. And finally, the changed polarity may allow the fluorine compound to adhere more firmly to the enzyme than would a uracil compound. Thus, we predict that the 5-fluorouracil, just like the uracil, will attach itself to a sugar phosphate residue, and then fit into the same place on the enzyme the uracil would have used. But once in place, the strong bond to fluorine will prevent the enzyme from replacing the fluorine with a methyl group. And finally, we predict that the strong polarity of the 5-fluorouracil will cause such strong adherence to the enzyme that the normal uracil compound will be prevented from reacting, and that, with the enzyme blocked, the essential chromosomal material cannot be made, and the cell will die. Let's test this prediction in the laboratory. Here are 2 cultures of cancer cells. Those on the left have been treated with 5-fluorouracil. Compare the action of these cells to that of the untreated cells on the right. The untreated cells continue to show rapid growth. The 5-fluorouracil treatment is seen to stop cell division, produce cellular enlargement, and eventually contraction and death. But are we sure that the cancer growth is stopped in the manner we predicted? Instead of the 5-fluorouracil blocking the enzyme as we have suggested, and using this means to kill the cell, is it possible that the 5-fluorouracil is incorporated into the DNA, inhibiting its function? To determine where the 5-fluorouracil is acting, in the enzyme or the DNA, let's conduct an experiment on some human cancer cells grown in the laboratory. A sterile pipette is used to transfer the cells into a large flask, which contains a nutrient medium. This permits continued growth so that we will have enough cells for our experiment. The cells are kept in suspension by a magnetic stir and incubated for several days. Here is specially prepared 5-fluorouracil containing radioactive carbon-14. By tagging the 5-fluorouracil with these radioactive atoms, we can determine where the 5-fluorouracil goes in a human cell. We'll add a small amount of the radioactive 5-fluorouracil to the living culture. In about half an hour, these radioactive molecules will be incorporated into the cancer cells. The cancer cell suspension is poured into large cups to be spun in a centrifuge. The spinning collects the suspended cancer cells into a pellet on the bottom of the cup. The liquid is discarded. By this means, the cells from large volumes of the culture medium are finally concentrated into one cup. The cells are then resuspended in cold sodium chloride solution to dissolve the DNA. The low temperature retards enzyme action. This cell suspension is placed in a blender. The violent action of the blender fragments the cells to facilitate dissolving DNA. After storage overnight in a refrigerator, the DNA dissolves, and then the solution will be spun very rapidly in a centrifuge. This spinning action separates the residual cell proteins which contain the enzymes. A high-speed centrifuge is required because the large size of the DNA has made the solution viscous. After the centrifuging, the liquid fraction contains the soluble DNA. The pellet contains the undissolved residue. According to our theory, if the 5-fluorouracil is incorporated into the DNA molecule, radioactivity should be detected in the liquid. Otherwise, it should be found in the pellet. The next step is to concentrate the DNA. The liquid is drawn off and injected into alcohol, which precipitates the DNA. These threads of DNA material can be readily collected on a glass stirring rod. A sample of the DNA fraction is removed for measurement of its radioactive content. The protein pellet is retrieved from the centrifuge tube. The meter shows that there is no radioactivity in the chromosomal material. However, the protein fraction does show radioactivity. The experiment proves that the radioactive 5-fluorouracil is not incorporated into the DNA. Though the chemistry is oversimplified as portrayed here, it does show that the structural similarity of 5-fluorouracil to uracil is of crucial importance in its ultimate effect, the enzymatic synthesis of genetic material. 5-fluorouracil is currently one of the most effective chemotherapeutic agents for treating certain types of skin cancer and colon cancer. About 40 anti-cancer agents are in clinical use today. For example, chemotherapy with methotrexate can lead to complete remission for 97% of children treated for acute lymphocytic leukemia. But we need even more effective and less toxic chemicals, especially for lung cancers and brain tumors. In the past, most medicines have been discovered by trial and error, even by accident. Aspirin, the most widely used medicine in the world, was used for almost 100 years with no idea as to how it reacted in the body. Modern techniques are allowing us to learn about living systems at the molecular level and what changes cause loss of health. Now some mechanisms are understood. We are beginning to be able to design molecules the living system needs for recovery.
what we're going to do is we're going to go into the computer lab. Now we're going to get a little introduction to quantum mechanics, quantum mechanical calculations.
This is not one for O2. Everything's normal. So I have to leave out the
I don't come on this.
No, all the orbitals are there.
Still,
It's
like,
just the
working with
ring,
people
the
is
sigma 2p
better
is
than
the
working with
pi,
different pieces of
the
glass.
drops here,
Okay.
the pi. So we go here above the sigma stuff.
Okay.
And then you have two options. You could do it with the core or without the core. You have to be very careful. Some books do it with a core, some books do it without. And so what happens a lot is people get it mixed up and they miscount the electrons. Like they think they're doing it for the core and the valence, but it turns out it's just the valence. So just be careful.
So they just ignore the 1s?
Yeah, right. And so you have to be careful which diagram you're using. You know, are you ignoring the 1s or incorporating the 1s? And that's going to change. You're either going to put the total electrons or just the valence electrons in there. So you just have to be careful, that's all.
So the order that I did it in would be okay for nitrogen or
for
lower?
nitrogen, carbon, right.
Okay.
It's no problem.
I don't know if this is a dumb question or if you know the answer, but in the video he was using like he had like cancer cells in a vial, right? So if he spilled that on himself, could the cancer cells interact with his body?
Well, there's different theories about, and there are different types of cancer. And so one type of cancer is a virus. As a virus, that would, then you could, obviously, you could contract the virus.
Okay.
But in this particular one, it must not be. Otherwise, I think you would take more precautions.
Yeah, because he wasn't wearing gloves.
Yeah, initially. Later on, he started wearing gloves, but initially he wasn't wearing any gloves. You're right. The same thing. I have a friend who's a biochemist, and she was working with prions, The prions are like mad cow disease, it's proteins, and that you could also contract.
Oh, that's scary.
Yeah, it's scary. So, I think that was, what they did. So I don't think that was contagious, at least, not. But if it was some kind of viral origin, then yeah, could be. So you'd want to watch out,
Okay. Yeah, that's pretty interesting. I didn't know that the structure. So structure is probably like way more important than maybe realize.
Oh yeah.
To this point.
Yeah, structure is and so this is why we want it, as accurate of a picture of structure as possible, because it matters. It matters how the electron cloud is distributed in the molecule itself, how it's going to interact with other species. So all this stuff is important to figure out,